Hello, everyone. We're going to have another lecture today. It's the respiratory system physiology. It's the unit 4, lecture number 2. Okay, physiology for respiratory system. And please read your textbook from page 840 to 854 before you watch this video. And also, I like you to work on the worksheet in your lab handout. Okay, something called the volume and capacity, that worksheet. So list out all the different terms for the volume here, and the capacity is basically is the questions related to this diagram you're seeing on my screen. So please do that before you start watching this video. Okay, so let me see if I can enlarge this. Okay, so first, the physiology part, we're going to have some terms. The first one, the tidal volume, is this yellow part in this diagram. Okay, so if you look at the y-axis, this is the volume, right? 1,000 ml to 6,000 ml. In your respiratory system, the volume you can hold the air. Okay, so here the yellow part, okay, if it goes up, that's inhale, goes down is exhale. That's the volume. All right, so the tidal volume is defined as at the quiet respiration, the air you inhaled in or exhaled out. This volume is called tidal volume. For a normal person, it is around 500 mLs. You do not need to remember this volume number here. If on the test I gave you a question, I will give you the volumes for each one. Okay. And then this pink part right here is called inspiratory reserve volume. That is the volume on top of the tidal volume. If you force yourself, you can inhale more air in. In this case, more muscles are involved, such as the scalene muscles, pec minor, serratus anterior, and also sternocleidomastoid. These muscles contraction will allow the thoracic cavity increase the capacity and allow more air to come in. So this part of the volume inhaled in on top of the tidal volume is called inspiratory reserve volume. Okay. Next one is expiratory reserve volume. That means on top of the quiet tidal volume you exhaled out. If you force yourself, if you contract the muscles like internal intercostals, rectus abdominis, and also like the oblique muscles, transversus abdominal muscles. If you contract those muscles, you can force more air out. Okay, so that's in this diagram is this blue part. It is called respiratory reserve volume. Okay, so you can push more air out on top of the tidal volume. Okay, and then this green part right here is called residual volume. It is about 1,200 ml. This is the volume stay in your lung. Okay, no matter how hard you try to force yourself to blow this air out, the residual volume, you cannot release it out until you die. Okay, so you probably heard about a dead person will have the last breath blow out air. That will be the residual volume. Okay, why do we have residual volume? There's a functional significance here. If you remember on the previous lecture, I told you there are some mechanisms to prevent the total collapse of the lung. One is a surfactant coating inside of the alveoli to reduce surface tension inside of the lung. And also another mechanism is in the pleural cavity, okay, watery fluid, because the water has charges on the surface, which will increase the surface tension to attract visceral pleura to the parietal pleura. That part will help to prevent the total lung collapse as well. So this is the third mechanism here, because we always have a small amount of air inside of the lung in the alveoli, and this part of the gas, the air, can help us to prevent the alveoli wall collapse from each other. Okay, so that's the residual volume. All right, so on this side is the capacity. Capacity is how much air the lung can handle. Okay, so the inspiratory capacity, that is the total volume you can inhale in. That is the tidal volume plus the inspiratory reserve volume. Okay. Functional residual capacity, that means this the air actually total air that you can blow out. Okay, that's the expiratory reserve volume plus residual volume. This vital capacity actually is more important in terms of function. So that is the total capacity the lung can handle. Okay, so the volume, that is the inspiratory reserve plus tidal plus expiratory reserve, not including the residual, because this part of the air cannot leave the lung unless you die. Okay, and then this is the absolute total lung capacity, which will include the residual volume here. All right, this slide is give you continued from the other slide, give you the capacities we just talked about. All right, next is a concept for dead space. All right, so we know when we talk about pulmonary ventilation, we're talking about air coming in or out of the lung. So in this part, we do not consider how much air is actually involved in a gas exchange. We just talk about air coming in or leave the lung. Okay, so if you think about the conducting zone we talked about last time, conducting zone is just the airway, allow the air to come in and out. There's no gas exchanges in that part of the tubular structure. So we call the volume in the conducting zone as anatomical dead space volume. That means these volume of air, even though they come into your respiratory system, but they're not involved in gas exchange. The volume for a typical person, average is about 150 ml. That is called anatomical dead space. There's another term, it's called alveolar dead space. Okay, so normally when we say, if the air flow inside of alveoli, then it should be able to do the gas exchange, because alveoli wall is very thin and there are many blood capillaries around. So gas exchanges can happen. But if there's something wrong with the alveoli. For example, TB patients, the alveoli damaged and they replaced, the simple squamous epithelial cells will be replaced by scar tissue. In that case, even though the air get into the alveoli, but still there's no gas exchange can take place because the gas cannot diffuse across the scar tissue. So in that case, the volume in those damaged alveoli will be considered as dead space as well. Okay, so that will be called alveolar dead space. So when you talk about the dead space, you should consider both the anatomical and alveolar dead space. All this volume from anatomical and alveolar dead spaces, these volumes of air, they are not involved in gas exchange. All right, so if we do the pulmonary functional tests, we use an instrument is called a spirometer. All right, so you can blow air into this instrument, and then there's a meter to measure the volume. Okay, so using this instrument, spirometer, we can actually diagnose either the obstructive or restrictive disorders. Okay, obstructive is basically the airway problem, resistance. Like say if you have asthma, all right, the airway constricted, so more resistance for air going in or out. That is considered as obstructive problem. If it's a restrictive problem, and that is the total capacity problem. Let's say if you have emphysema, for example, damaged a lot of alveoli. Okay, so not damage a lot of alveoli, so the total capacity in the lung is reduced dramatically. That is called restrictive problems. Okay, so there are a few important tests you probably should know. Okay, one is the total minute ventilation. That is the total amount of air flow into or out of respiratory tract in 1 minute. So if you have, like say you're sitting, quietly, each breath you're going to have the tidal volume, right, in or out, times the rate, how many breaths you have in 1 minute, then you will have this total minute ventilation. Okay, next one is called a forced vital capacity, gas forcibly expelled after taking a deep breath. Okay, so the air coming out. Okay, so it's no time limit. You just try to blow out as much as you can. So this is the maximum volume of air you can blow out. That it will can be used to test restrictive problem. If your lung for some reason is damaged, then it holds less air in, then this number will be small. The third one is called the forced expiratory volume. This one is the amount of gas expelled during specific time intervals. So normally it's in the first second. So basically you take a deep breath, inhale as much as you can, and then you start to blow, you start to measure and also time how much air you get out in the first second. All right, so this one is used for diagnose the obstructive problem. I say if you have asthma, your airway is constricted. So in first second, the air you blow out should be less than a normal person. Okay. Next term, this one is important concept. It's called alveolar ventilation. We talked about the pulmonary ventilation last time. The air in and out is called pulmonary ventilation. Alveolar ventilation is more important because this one gave you a tell you how much air actually involved in gas exchange. So this is more important in terms of study the functions. Okay, so this one is called alveolar ventilation rate equals the how many breaths per minute times, look at this part, okay, tidal volume is each breath, the air in or out at a quiet situation. And you're not using this tidal volume, but you use the tidal volume subtract the dead space volume. So actually, after this one, tidal volume minus the dead space volume, this is the volume actually involved in gas exchange, right? So this volume times how many breaths per minute, that will give you the alveolar ventilation rate. So to explain this concept, let's look at this example here. There's this chart gave you different patterns of breathing, okay, breathing patterns. This one is a quiet breathing, okay. This one is very deep but slow breathing. This one is a shallow but really fast breathing. So three different patterns. So if you look at here, for first one, quiet, we know the tidal volume is about 500, and then the breaths per minute is about 12, okay? The dead space is the anatomical dead space volume is 150, and then the pulmonary ventilation, the total volume in or out in 1 minute is 6,000 mLs. Okay, but if you look at the alveolar ventilation, it's about 4,200. That means out of this 6,000, 4,200 ml of air actually get into alveolar and then involved in the gas exchange. Okay, now if you look at the second one, deep but slow breathing, you can try that. Okay, deep but slow. So each time you inhale, you're going to inhale more air in, and then the breaths per minute is lower. The dead space does not change. That is the conducting zone, right? And then the total amount of air in and out or out in 1 minute is the same as the top one, okay? But if you look at the alveolar ventilation, there's a difference. Okay, now look at the third one here. Okay, shallow but fast, kind of like the dogs, you know. Okay, really fast but very shallow. So the tidal volume is lower than normal, 150, and the breaths per minute is much more than normal. Okay, the dead space does not change, right? And pulmonary ventilation, the total volume in or out in 1 minute is still 6,000 mLs. But how much air get into the alveolar for gas exchange? 0. You see this big difference now? Okay, this is why probably you have this experience, you know, especially ladies, you know, when we get emotional, we start to cry. When we cry, we start to have this shallow but fast breathing, okay, like that. And then you will feel like you have lightheaded and you may even pass out. The reason for that is now you don't even have any you do not have any volume of air inside of your alveoli for gas exchange, so you pass out from lack of oxygen. Okay, all right, so this chart, I just want to show you that alveolar ventilation is more important in terms of function. Okay, so now we can talk about the gas exchanges in the body. Okay, the terms, a few background you need to know, but this one I'm not testing you, just give you an idea. Okay, Dalton's law means the mixture of the air have a total pressure, like atmospheric pressure. This pressure equals to all partial pressure for it's the sum of partial pressures for all the different gases in this mixture. For example, in our air, we say the atmospheric pressure is 760 millimeter mercury. Millimeter mercury is the unit to measure the gas pressure. All right, so this is 760 millimeter mercury equals to all the partial pressure, all the gases, partial pressures to sum up. So like say oxygen partial pressure plus nitrogen partial pressure plus the partial pressure for CO2, partial pressure for carbon monoxide, and all the gases, hydrogen, hydrogen, and so on. So if we put all these partial pressures together, add them up, that should equals to 760. Okay. And then in the air, okay, the most gases, the will be nitrogen, oxygen, and carbon dioxide. These are the most, nitrogen is the first one, and then oxygen, and then carbon dioxide. Okay. Henry's law is talking about each gas has a ability to dissolve in the liquid. However, how much the gas can dissolve in the liquid it's a property of each gas. Okay, so they're different. For example, CO2, carbon dioxide, can dissolve more in the water than oxygen. Okay, so that is the property of each gas. And also, if you change the pressure or temperature, you can affect the solubility of the gas in the liquid. So this one you probably will have a experience. Let's think about a soda can. All right, so soda can, how can you make a soda? It's basically water, and then you apply a lot of pressure and push CO2 into water, dissolve in the water. That is a soda, right? So if you don't apply that pressure, you only have a certain amount of CO2 dissolved. But when you put more pressure on it, you can dissolve more CO2 in water. Okay, so that's the first part. The second part, if you change the temperature, you can change the solubility. So if you have a soda can, you put on outside in the summer day for an hour, and when you open it, you will know all these gas try to come out from the water, right? So higher temperature, less solubility. All right, so this one gives you an idea how much each gas in the air mix. Okay, nitrogen is the most, 78%. Okay, oxygen 21%. Carbon dioxide is only 0.04%, but still we consider these three are the most gases in the air mix. Okay, the rest of them, like carbon monoxide, hydrogen gas, they're much less than even the carbon dioxide. Okay. All right, now we can actually talk about the gas exchange. Do you remember this word, external respiration? This is the gas exchange between the lung and blood capillaries. How about internal respiration? Is a gas exchange between blood vessels and tissue cells. Okay, in between the external and the internal respiration should be the gas transport in blood. Okay, this is a very useful or diagram, have a lot of information here. Okay, the top part is the external respiration. So you see this is the lung alveoli, this is a capillary in the lung, capillary beds in the lung. So there's a gas exchange over here. And this part is the internal respiration between the cells and blood capillaries. In between, they're just a gas transport in the circulatory system. Okay, but you need to know more details here. Okay, in the alveoli, if you see the partial pressure for oxygen is 104. 104. Okay, if you see partial pressure for CO2 is about 40. These two numbers you probably should remember, okay, because later on you know to understand the mechanism you need to know these numbers. So one more time, in the lung, when you actually inhale fresh air in, the partial pressure for oxygen is about 104. The partial pressure for CO2 is about 40. Okay, now in the blood in the pulmonary area here before gas exchange, we know this blood is oxygen poor blood. Okay, so in this blood, look at the oxygen here. Oxygen in the blood is about 40. Partial pressure is about 40. CO2 is about 45. So now let's just look at the oxygen. In the blood, partial pressure is 40. Outside in the lung, partial pressure is 104. Now think about oxygen will flow from where to where? It's from an air sac to the blood or from blood to the air sac? Definitely from high to low, right? From higher pressure point to the lower pressure point. So oxygen will diffuse into the blood. Same principle, if you look at the CO2 in these oxygen poor blood, it's 45. and, and in the air sac is about 40. So the CO2 will move from the blood to the air sac. Okay, so this is external respiration. If you look at the internal respiration here, okay, look at the blood. Excuse me. Let me have some water. Okay, so if you look at this oxygenated blood, oxygen-rich blood, PO2 is about 104. Partial pressure for CO2 is about 40. If you look at the tissue cells, the partial pressure for oxygen is about 40. Okay, now you understand. So the oxygen will leave the capillaries and then get into the cell. And same principle, CO2 will leave the cells and get into the blood. Okay, all right, so this is a diagram you probably should go over by yourself again. Okay, and the next concept is called ventilation perfusion coupling. This is the mechanism for gas exchange in the lung. Okay, ventilation talking about the air in and out. Okay, ventilation talk about the movement of the gas. Perfusion talk about the movement of blood. Okay, so ventilation is for gas, perfusion is for blood. So let's look here. This yellow part representing the alveoli. Okay, and then here, this is blood capillaries around here, around the alveoli. Okay, so the if there are less air get into this alveoli, for some reason, not very much air into this part of the lung, then that's the term we should use is the less ventilation in this part of the lung. Then what's going to happen to the blood vessels? You see, the capillaries are constricted to minimize blood coming to this area. So to reduce the perfusion, blood flow to this area. This is a very efficient mechanism. So if you don't have a lot of air coming in, then there's no use to send a lot of blood to this area because you don't have very much of gas for gas exchange. Does that make sense? All right. So if you look at this one, if there are a lot of air coming in to this area in the lung, then the blood capillaries, what did I do? Then the blood capillaries will dilate to allow more perfusion, allow more blood coming to this area to do gas exchange with the lung tissue. So this is called ventilation perfusion coupling. Okay, ventilation and perfusion, they always go side by side. Higher ventilation, higher perfusion. Okay, lower ventilation, lower perfusion. All right, so we talked about external and internal respiration. Okay, now the only thing left is kind of like in between is the gas transport. Okay, so when we talk about a gas transport in the blood, we talk about oxygen transport and CO2 transport. Okay, so first is the oxygen transport. Oxygen combined with hemoglobin. Okay, if you remember, hemoglobin structure has a heme group inside. Okay, each hemoglobin can carry 4 oxygen molecules. Okay, so oxygen can bind on the hemoglobin and travel that way. Oxygen can also dissolve in plasma. Remember, we said each gas has a solubility in liquid. So some of the oxygen can directly dissolve in the plasma and travel that way. But the majority of oxygen will travel in blood in the form of binding to the hemoglobin. So if you look at this diagram here, yellow part is alveolus, and then this is a capillary, blood capillary. So if we enlarge this, all right, yellow part still is alveolar sac or alveolar air sac, and on this side is blood capillary. So you have red blood cells. Of course, the hemoglobins are inside of RBCs, right? And also you have plasma here. So oxygen can dissolve, can diffuse from air sac into the blood. A small amount will dissolve in the plasma. Most of them will travel inside of RBC and bind to the hemoglobin. So HB here represents hemoglobin. So once the hemoglobin bind with oxygen, we write it this way, HB dot oxygen. Okay, and then this is the external respiration part, right? This is the internal respiration. So now you see this is blood vessel and tissue cells. Okay, we know at this part, the oxygen will unload. Okay, that's the word. That means the oxygen will dissociate from the hemoglobin, and now it's a free oxygen, right? That's called oxygen unload. And oxygen will diffuse out from blood and get into the tissue cells. Okay, so this part of movement is a simple diffusion. It's just because there are more oxygen in the blood, less in the tissue. So oxygen will go from higher pressure point to the lower pressure point. Okay, so I should spend some time on this curve. This is a very, very important curve. Everybody needs to understand this. This is called hemoglobin saturation curve, right? On the y-axis is the percentage of oxygen bind to the hemoglobin. So the highest will be 100% binding. On the x-axis is the partial pressure for oxygen. Partial pressure for oxygen. Do you remember the partial pressure for oxygen in the alveoli? 104. Yes? Okay. So that's why it says it's around the lung right here. So if you look at this, if the partial pressure for oxygen is around 100 or 104, look at this hemoglobin saturation is almost 100%. It's not 100%, but it's very close to 100%. This is what we call oxygenated or oxygen-rich blood. Okay. Now think about in the tissue cells, the partial pressure for oxygen is, do you remember? Around 40. See here? Tissue. PO2, oops. A partial pressure for oxygen is about 40 right here. So if at a partial pressure, oxygen partial pressure at 40, if you look at a hemoglobin saturation here, it's around what, 70? Yeah, it's around 70%. So that means in the tissue area, in the blood, still 70% of oxygen are associated with the hemoglobin. This is what we call oxygen poor blood. So now you know when we talk about oxygen rich, oxygen poor is only relative terms, okay? Oxygen poor blood still have 70% of oxygen associated with hemoglobin. Or you can say oxygen poor blood still have 70% of oxygen loaded on the hemoglobin. Okay, so we have a huge reserve, right? Even oxygen poor blood, we still have 70% traveling in the blood. Okay, so from here to here, to zero, there's a huge reserve. Like say if you this is a good protective mechanism. If you suddenly get into the situation you need to have more oxygen, then you definitely can unload more oxygen from hemoglobin. So this is called a hemoglobin saturation curve. And if you look at this curve, once the we should say from pressure, oxygen pressure 20 to 60, in this range, you change the pressure a little bit, you can change the saturation a lot, because this part of the curve is steep. Okay, that means you change the pressure a little bit, you can change the saturation a lot. But once you get above 80, oxygen pressure above 80, so from here to here, let's use an example. At partial pressure 80, the saturation is about 95%, right? 95% over here. But around 100, partial pressure for oxygen around 100, this saturation is around 99% maybe. So it's not a huge difference. The only maybe like 4% difference. But if you look here, okay, if you change, let's say from 30, okay, partial pressure 30 here, the saturation is about 58%. But if it's around 40, think about the partial pressure here, it's about 70. Okay, 58, 70. The difference is much, much bigger than from here to here. Did you get what I'm saying? Okay. So that is called, this curve is called a hemoglobin saturation curve. And the hemoglobin binds to the oxygen on the heme group. Okay, so each hemoglobin molecule has four sides to bind to the oxygen. And they work in the fashion, like say, if one hemoglobin bind to one molecule, one oxygen molecule, then the rest of the hemoglobin will all bind with one oxygen. So in that case, we can say it's 25% saturation, all right? And then if the second one bind to the hemoglobin, then the rest of the hemoglobin will have the second one bind to it. So then it's a 50% saturation. So in this case, you can see most of the hemoglobins will have 3 oxygen occupied on this hemoglobin molecule. Then it's a 75% saturation. And in this one, okay, all 4 sides are occupied by oxygen. In this case, we say it's close to 100% saturation. Okay. Let me skip this one. Okay, so now hemoglobin and oxygen, this curve is very important because it will tell you at what oxygen pressure, how much oxygen can dissociate from hemoglobin and get to the tissue cells, right? And then we know factors can definitely affect this curve, okay, like a temperature, like hydrogen ion concentration, which is the pH, right? And CO2 pressure, and the BPG. This one is a metabolic waste, a product from chemical reactions, okay, and also hormones. Oops, too fast. Okay, so let's use this curve as example. So the blue one is normal body temperature, okay? So this one, let's see if I can annotate. Let me see if I can get my, no, I can't. Where did my, darn it. That's funny. Okay, I just used the arrow here. Okay, so this is a 37 degrees. That's the normal body temperature. So at this temperature, all right, let's say at a 40 PO2 here, if you draw a line here, 40 PO2, draw the line, get to this blue, and then cross to the saturation. Let's say it's about a 70% saturation. So that is what we call the oxygen poor blood, okay, still have a 70% saturation. But if you increase the body temperature from 37 to 43 Celsius, okay, now you see this curve, this purple one shifted to the right. This side is right. So the curve shift from this blue one to the right of the blue, which is this purple one. What does that mean? That means, like say, if we look at partial pressure, again, is a 40. Now, if you look at the saturation here, it's only 60. Did you get it? Before, at PO2, partial pressure for oxygen at 40, the saturation is about 70. But if you increase temperature, then this curve is going to shift to the right, and then you use the same principle. If you draw a line from partial pressure at 40, and then see the saturation is only 60. That means if you increase temperature, more oxygen will unload from hemoglobin and get into the tissue cells. This is what we want because higher temperature, that means faster, in general, faster chemical reactions. Then you need to have more energy. You want to have more oxygen in the cells to make ATP. Okay, so same idea. If the temperature drops, this yellow one, okay, again, then this curve is going to shift to the left, and then at the same partial oxygen pressure, the saturation will be above 80. Okay, that means if the temperature is low, your metabolism is slower, and then you don't need to have that much oxygen, so you will not unload that much oxygen, and then more oxygen will be still associated with hemoglobin. Okay, so next one is the hydrogen. Remember, free hydrogen ion concentration determines the pH. The higher the hydrogen, the lower the pH, right? Okay, so maybe let me explain the CO2 first. Maybe this is easier, okay? Let me see if I can use my whiteboard here. Okay, so let's use the whiteboard. Okay, CO2 plus water can make this acid. All right, and then this acid can partially dissociate into bicarbonate ion and hydrogen ion. Yes? Okay, this is a positive sign here. Okay, so if you increase CO2 concentration, the reaction turns to this direction. Okay, then you're going to have more hydrogen ion concentration. Does that make sense? Everybody? Okay, this is a chemical reaction you learned in the chemistry, and this arrow means the reaction can go in both ways. It solely depend on which side has more materials. So if you have more CO2, then the reaction will go in this direction. Okay, so when do you have more CO2? That's when you have a higher metabolism, because the CO2 is the product of metabolic reactions, is the waste. Okay, so if you have more CO2, what's going to happen to this saturation curve? Okay, let's take a look. Okay, so CO2 right here, all right? If you have more CO2, then your pH, let's see if I have another curve here. Oops. Why does it not move? Eh? Either doesn't or move too fast. Okay, so if you have more CO2, what is going to happen to this curve? Think about it for a minute, okay? And I don't have a curve on my slide, but if you can have your book on page 850, the second curve, you see the red one is when you have more CO2. So basically the curve is going to shift to the right. What does that mean? That means more oxygen will unload from hemoglobin. Okay. If you have more CO2, that means the metabolic rate is higher. then that's why you have more CO2, the waste, right? If your metabolic rate is higher, then you want to have more oxygen, okay, to support these chemical reactions. So this makes sense, it's similar to the temperature. Higher CO2, higher metabolic rate, higher temperature, higher metabolic rate. In both cases, you're going to have more oxygen. You want to have more oxygen unloaded from hemoglobin and get to the tissue cells. Okay, same thing for the BPG, because this one is metabolic waste. The more of this, that indicates the stronger metabolism, and then you want to unload more oxygen from hemoglobin. Okay, now hydrogen. Okay, so if you look, we did say higher hydrogen concentration. That means what? Lower pH, right? So if you increase CO2 concentration, are you going to generate more hydrogen ions by the chemical reactions I just showed you? Increase CO2, generate more hydrogen ions, then you have a lower pH, right? So increase CO2, increase hydrogen ion concentration, lower pH, and the curve is going to shift it to the right. Okay, so I know it's a lot of information, and if you feel like you do not grasp all of these concepts, you may want to stop here and go back to your book, read from 848 to 850 before you continue, okay? Alrighty, so we're still talking about oxygen transport, right? So we talk about oxygen combined with hemoglobin and transport in RBC. This one is giving you some ideas for CO2 transport, okay? So oxygen transport is here. CO2 also transported in blood, but CO2 here you can see CO2 can diffuse and into the RBCs and bind to the hemoglobin. If you remember this, CO2 combined with the hemoglobin, but it's not the same site for oxygen. All right, it's bind to this different part on the hemoglobin. And CO2 can also transport it in blood in the form of bicarbonate ions. We'll talk about that. Okay. Also, CO2, some of the carbon dioxide can dissolve in the plasma. All right. So let's talk about the CO2 transport. Okay. So if you look at here, this is blood capillaries on this side. These are the tissue cell. So this diagram representing the external respiration or internal respiration. Internal, right? So it is between the cell and blood. Okay, so you see there are 4 arrows here, 4 arrows, okay? These 2 are in the plasma. These 2 are into the RBCs. The biggest arrow representing most of the CO2 will transport this way. So we'll start from this one. So CO2 generated by tissue cells diffuse into blood and then directly into RBC. In the RBC, CO2 and the water combine to make this acid, bicarbonate carbonate acid, and then this carbonate acid will dissolve partially into bicarbonate ion and hydrogen ions. This reaction requires an enzyme, is carbonic anhydrase. This enzyme is only in the RBC, not in the plasma. Okay, so because of these anhydrase, this reaction can take place really fast. So CO2 can be converted to the form of bicarbonate ion and hydrogen ion. Okay, so now in the blood, if you have a lot of bicarbonate ions accumulated, it's going to affect the speed of this reaction because you build up the products on this side. For that reason, bicarbonate ions can be transported into plasma. This part is the plasma. Okay, to balance the charges, the chloride, the same negative charges will be transported into the RBC. So once the bicarbonate ions get into the plasma, it can quickly dissolve into the plasma. All right. So because it's a charged particle, it can dissolve in water and then travel in the form of bicarbonate ions. It dissolved in plasma. So this mechanism, we call it, the CO2 will transport in the blood in the form of bicarbonate ions. Okay, so as long as you remove bicarbonate ions into the plasma, then you can continue this reaction. CO2 will continue to be converted to the form of bicarbonate ions and dissolve in plasma and travel in blood transport, blood circulation. Okay, the second line right here, the arrow, CO2 diffuse into blood and then into RBC, and then combine to the hemoglobin to form a carbaminohemoglobin. Okay, so if you remember the previous slide, we say about 23% of CO2 can travel this way. Okay, so just the CO2 binds to the hemoglobin. And then the third arrow here, CO2 diffuse into the plasma and directly dissolve in the plasma. This one probably about 7%. Okay, and then the thinnest arrow here, very small amount of CO2 will get to the plasma, bind to the water, and then convert it to the bicarbonate ions. It's the same reaction, but in the plasma because there's no enzymes there. So this reaction is very slow. Okay, so we can say there are 4 ways for CO2 to transport in blood. This is internal respiration part. This is the external respiration. So again, this is the blood capillaries. This is the air sac, alveolus. All right. So now you see this is the biggest arrow. So CO2 diffuse from RBC to the air sac. And the reaction is the reverse one for the previous slide. Okay, bicarbonate ions bind with hydrogen and convert it to the CO2 and the water. As long as you continue to remove CO2 to the air sac, you continue to blow out the CO2, and this reaction will continue. All right, more and more bicarbonate ions will be converted to the form of CO2 and diffuse out into the alveoli. Okay, and then this one is the CO2 will dissociate from hemoglobin and then diffuse into alveoli. Okay, same for these 2 parts, just reverse reactions from the previous slide. All right, so the last part is about control of respiration. So this part I'm going to, I think I'm going to go easy on you. So I want you to know the main things. One, there are different levels of respiration control. The rate and the depth of the breathing, different levels. The most important part is in the medulla. Okay, medulla respiration centers, and then in the palms we do have a respiration control center, which will continue to monitor the function of the medulla respiration centers. Okay, and then higher level in the cerebrum cortex, there are higher control centers which can monitor the respiration as well. So normally when you take a breath, you don't need to think about it. It's kind of like autonomically controlled, right? That is the medulla's centers for that. But you can definitely control your ventilation in a certain degree. Like say, if you sit here and say, okay, I want a deeper breath, then definitely you can inhale more. That part is directly controlled from a cortex, okay, cerebral cortex. So if you think about the Superman, the Christopher Reeve, okay, he was injured at the medulla part, totally damaged his respiratory center in medulla. So he lost it, the autonomic part of control for breathing, but his cortex was working well. So during the day, when he was still alive, during the day, he actually could control his breathing voluntarily. You know, when he was talking to someone, he would say a sentence and stop, inhale, and then continue talking, and stop, and inhale, and continue talking. So that part was from cortex. And the night, because his medulla was damaged, he couldn't, have autonomically controlled breathing. Then at the night, he had to have a breathing machine. He had to use a breathing machine. Okay, so that's pretty much what I want you to know from this slide. And okay, you can read this part. Okay, and then this slide, I want you to know that normally, okay, we have chemoreceptors which can sense the changes for oxygen and the CO2 concentrations. We have peripheral chemoreceptors in the big arteries, and also we have a central chemoreceptors inside of the cerebral spinal fluid, if you remember that. Okay, so normally it's the CO2 concentration determines your respiration pattern, how fast and how deep you breathe. Okay, so normally it's a CO2. Unless your oxygen is very, very low, or unless the CO2 sensors are not working well, then the pattern can be controlled, switch to the oxygen controlled pattern. Okay, so for a normal person, changing breathing pattern is determined by CO2 concentration. If you have a lot of CO2 in your body, then you will change the breathing pattern, try to blow out more CO2. Okay, but if you have a COPD, for example, you have a chronic lung problem, then the CO2 sensors will get desensed. Okay, it's not going to be as sensitive as normal. All right, it's not respond to the change of CO2 concentration that much. In this case, the pattern control, respiration pattern control, will be switched to oxygen sensors. Okay, that's all I want you to know for this part. Okay, and then you can read the rest of the slides. I think I'm going to stop here. All right, the rest of the slides I will not test you on, since this online thing, I'm still working on that, and I don't want to give you too much information in one lecture. So the rest of the slides for if you print out the lecture notes, you can see there are a few more slides here. You can read it through, but I'm not going to test you on that. Okay, so this is the end of the second lecture. Okay, next time we're going to start with the urinary anatomy. Okay,